Hello, my name is Mariana Byndloss, and I'm an assistant professor at Vanderbilt University Medical Center. And today I'm going to talk to you all about the work in my lab, focusing on how disruption of microbiota and intestinal epithelial interactions are key drivers in childhood obesity. The main focus of my lab is really to try to understand how the host and the gut microbiota work together to prevent disease. We are particularly interested in understanding how metabolic interactions between the gut microbes living in our gut and the intestinal epithelium prevent disease, but we're even more interested in understanding what happens when these beneficial interactions get disrupted, specifically by environmental factors such as diet and antibiotics, and how that can lead to non-communicable diseases such as obesity, diabetes, and colorectal cancer. We became very interested in obesity, in particular childhood obesity, because it's known that about 20% of the school-age kids in the US are obese. And if you look here where we are in Tennessee, this number is even higher, leading to about 20 to 24% of kids. So it's really a pressing need to try to understand the reason why behind this obesity epidemic. But we also know there are specific risk factors that may promote obesity. One of them is consumption of this Western style high fat diet during childhood. But another one is the use of antibiotics, specifically during early life. But when we're studying these risk factors, we usually tend to focus on studying one of them individually. And that makes sense because it's much easier to determine cause and effect when you're exposing individuals to just one risk factor at a time. However, if you're looking at what's happening to the kids in our country, you actually see that in the top here, in the states in which kids are more exposed to Western style high fat diets or are in food desert and have decreased access to healthy diets, are the same states that have an increased rate of antibiotic prescription. So because of that, when we started this project, we started really thinking of a simple question, which would be, what happens if we expose young individuals to more than one risk factor at the same time? In this case, we wanted to understand what happens when you expose young individuals to a combination of a high-fat diet and antibiotics at the same time. And this work was led by my very talented graduate student, Katie Shelton. So to do this, what we decided to do is base our work in Martin Blazer's group's work and actually expose these animals, so young animals from weaning age at 3 weeks of age until they reach sexual maturity at 8 weeks of age to different treatments. The first one is exposing them to a control low-fat diet. We also exposed them to antibiotics, and in this case, we used a constant low dose of antibiotics, so subclinical dose of penicillin. We also exposed them to a high-fat diet, and we exposed them to a combination of this high-fat diet and antibiotic treatment. And we also, this is important to note, is that we observed the same results that I'm going to show you here. If instead of using the subclinical dose, we actually did pulses of clinical doses of penicillin. So suggesting that the effects are similar whether you're exposed to this chronic subclinical doses or if a young individual is exposed to more than one clinical dose of antibiotics. And then we wanted to understand what was happening to these animals when they reach sexual maturity. So if you see here in the dark blue in our hands, exposure to antibiotics alone did not increase weight gain in the animals. However, if the animals were exposed to antibiotics in the background of high-fat diet, they show increased abdominal fat. And the same thing was true for increased weight gain, increasing fasting glucose levels, and increasing fatty liver disease. So this data suggested to us this early life exposure to more than one risk factor increased or accelerated obesity and obesity-related complications. But the question here is why? Why was this happening? So you can imagine that there's multiple reasons why this phenotype could be happening, but my lab in particular is interested in understanding what is happening to how the gut microbiome may play a role in this phenotype. And this is important because we knew from other work that antibiotic treatment disrupts the gut microbiome. High-fat diet alone exposure also disrupts the microbiome. But we were not sure what was happening to the microbiome in animals exposed to both high-fat diet and antibiotics. And what is important here is that we decided to focus in a different segment of the intestines, which is the small intestine. And the reason is because most of the nutrient absorption, specifically lipid absorption, and interactions between the microbiome and lipid metabolism is happening in the small intestine. And also, the small intestine and the small intestine microbiome are really understudied sites of very important host microbiome metabolic interactions. So because of that, what we decided to do is characterize the small intestine microbiota of those young animals exposed to different treatments. And what we saw here is that exposed to antibiotics alone did not alter the composition of the microbiome that much. However, exposure to a high-fat diet significantly decreased the diversity of the microbiome and led to an increase in lactobacillus in the small intestine. However, this increase in lactobacillus was reduced in animals receiving antibiotics. And we did additional experiments that I don't have time to show here to demonstrate that actually these lactobacillus species become more susceptible to the antibiotics in the presence of a high-fat diet and bile acids, suggesting that the high-fat diet maybe increased susceptibility of these microbes to antibiotics. But the key point here is that I wanted to remember is that lactobacillus become depleted from the small intestine microbiome of animals receiving a combination of a high-fat diet and antibiotics. Since we're interested in microbiome intestinal epithelial interactions, we next wanted to see what was happening to the small intestine epithelium of these animals. And important here is that we shift to understanding what was happening into the epithelium in the context, in the background of a high-fat diet, because that's where we saw our weight phenotype. And the RNA-seq data revealed that multiple genes related to lipid transport and metabolism were increased in animals receiving the combination of antibiotics and a high-fat diet. And actually, we could also see that the epithelium of these mice accumulated more triglycerides. And if you did a lipid challenge, so if you expose these animals to olive oil, and you looked at triglycerides in the serum after, you could see that there's increased triglycerides in the serum of the animals exposed to a combination of risk factors, suggesting that these treatments were really altering how the epithelial was handling fat and leading to increased circulating lipids in young animals. So all this data together made us think of key regulators of lipid metabolism, and it made us think of peroxisome proliferator receptors because they're key regulators of lipid metabolism. And those nuclear receptors have also been shown to be regulated by microbiome and microbiome metabolites. So we know from our work and others that short-chain fatty acids produced by the gut microbiome activates the peroxisome proliferator receptor gamma. So it's possible that other unidentified metabolites could be doing the same. And we also knew that antibiotic treatments deplete these microbes in the large intestine, and this is in the large intestine context. They deplete these beneficial microbes and deplete those metabolites, leading to reduction of PPAR-gamma function in the intestinal epithelium. So because of that, we wanted to understand whether this combination of a high-fat diet and antibiotics had any impact in PPAR-gamma function in the small intestine of our mice. And the simple answer to this is yes. So if you look by immunohistochemistry and we did other assays, we see PPAR-gamma activation and PPAR-gamma function was decreased in the intestinal epithelium of these mice. And we confirmed that this was the case by also doing some genetic-based experiments that I don't have time to show here. But we knew from these experiments that PPAR-gamma signaling is required for protection against antibiotic-induced obesity during early life. So that led us to our hypothesis up to now is that during early life, you're exposed to a high-fat diet. There's members of your small intestine microbiome somehow protect you against adiposity by activating PPAR-gamma. But when you're exposed to more than one risk factor at the same time, those beneficial microbes are no longer present, leading to decreased expression and function of PPAR-gamma in the epithelium, leading to adiposity. But the question here is, how is this happening? So to try to answer this question, we went back to our animals and we isolated the lactobacillus strain from these young mice, which in this case was Lactobacillus murinus, because we thought that this was maybe the protective microbe since it was depleted from animals receiving antibiotics. And what we decided to do is use a germ-free model in which we colonized young, so a 3-week-old germ-free mice with a microbiome that has lactobacillus and a defined microbiome that does not have it. And we exposed these animals to a high-fat diet. And what we saw is that animals that had lactobacillus in the small intestine microbiome were protected against high-fat diet-induced obesity, really suggesting to us that this was a protective microbe. But we wanted to understand if this was really due to changes in PPAR-gamma activation. So what we did, we moved into an in vitro assay to try to understand whether lactobacillus could activate PPAR-gamma in intestinal epithelial cells. So what we did, we exposed these intestinal epithelial cells to lactobacillus. And what we saw here, you can see in the immunofluorescence, is that you see increased PPAR-gamma nuclear translocation, which is a sign of PPAR-gamma activation. And what's more interesting is that we knew that this only happened if the lactobacillus was alive, and that if you use a lactobacillus supernatant, so if you use just the products of this microbe, the phenotype was the same, suggesting to us that it was probably some metabolic component, metabolite from lactobacillus growth that was activating PPAR-gamma in the intestinal epithelium. So we wanted to understand what this potential metabolite was. So to do that, we went back to our early life model, and we actually obtained small intestinal content from our animals exposed to a high-fat diet or a high-fat diet and antibiotics, and we performed untargeted metabolomics. And what you can see here, just by this heat map, is that really exposure to even low doses of antibiotics completely changes the metabolic landscape of the small intestine content. And you can see the multiple metabolites are impacted. So we did a lot of screening that I'm not going to discuss today, but we came upon one particular metabolite of interest, which was phenylactic acid, or PLA. And the reason is because PLA was significantly depleted from the small intestine content of animals receiving high-fat diet and antibiotics. And this metabolite is of interest because it turns out that it's present in fermented foods such as kimchi, which have been shown to have an anti-obesogenic effect. And what's even more interesting is that my lactobacillus that was shown to be protective produces high levels of PLA. As you can see here, measurements of PLA in Lactobacillus supernatant. So we wanted to really see whether PLA could be protective by activating PPAR-gamma intestinal epithelial cells. So to do that, we repeated the experiment that I had shown you before in vitro. And what you can see is that indeed PLA activates PPAR-gamma to similar levels that we had seen in lactobacillus, suggesting that PLA could really be a lactobacillus-derived metabolite that activates PPAR-gamma in the intestinal epithelium. But this was very interesting, but I think the quick question for us was that, so we've identified this metabolite, but now we want to understand whether PLA can prevent this antibiotic-associated early life obesity in vivo, right, in our animal model. So to do that, we went back to our early life model and we supplemented PLA to the mice by giving it in the drinking water. And this is a data that I showed you earlier between the light red and dark red is that animals receiving antibiotics have increased abdominal fat. You also see that PLA does not have an effect in animals not receiving antibiotics. And the reason is because those animals actually already have high levels of PLA. But by the PLA treatment actually protects the animals from this antibiotic-induced adiposity by restoring PLA levels in the gut. We've done this measurement. And it actually also prevents weight gain and fatty liver disease in these young animals, which is very exciting to us. Leading us to this idea that during early life, when you have a high-fat diet exposure, members of the small intestine microbiome, in this case, Lactobacillus murinus, produces metabolites such as PLA that activate PPAR-gamma in the intestinal epithelium, preventing increased fat absorption and a secretion by intestinal epithelial cells, decreasing adiposity. However, if you're exposed to more than one risk factor at the same time, these beneficial microbes get depleted, leading to increased fat absorption and secretion by the intestinal epithelial cells, leading to accelerated adiposity. So this was a very interesting result to us. And what's even more interesting is that you can actually find PLA in the intestinal content of infants, so very young humans. And there's some data suggesting that PLA is actually more abundant in breastfed babies versus formula-fed babies. Really made us think that maybe this microbiome, intestinal epithelial interactions were relevant even before this after weaning period. Maybe it was even more relevant in the pre-weaning period. And made us think about how does this early life microbiome disruption may impact intestinal development and host metabolism during very early life. Thinking about this interaction between the small intestine microbiota, intestinal epithelium, and host metabolism during early life. So to do this, we actually changed our animal model, and we now moved to expose pups to antibiotics to cause a disruption much earlier in life. So in this case, we expose moms and the babies to antibiotics from right before birth until the first 3 weeks of life. And what we wanted to see is how is this impacting first the microbiome of the pups, with the focus on the small intestine, which is important here. So the first thing that we noticed is that between 2 to 3 weeks of life, there's a significant expansion of the overall number of microbes in the ileum. So at 2 weeks, those animals are almost germ-free. They have a very low level of microbes in the small intestine, and that changes by 3 weeks of age. And the overall number of microbes does not seem to be highly impacted by our treatment. However, when you look at the microbiome composition, you actually start to see some differences and loss in microbial diversity with a significant depletion of Clostridiaceae. But when we do additional analysis, we actually notice that two specific microbes bloom between 2 to 3 weeks. The first one is the Lactobacillus intestinalis that significantly increases by 3 weeks, and this microbe actually gets depleted by antibiotic treatments. And the second one is segmented filamentous bacterial, or SFB, which also blooms a lot in the small intestine by 3 weeks of age, but is very much depleted in animals receiving low-dose penicillin. It's almost like these animals are never colonized with SFB. So we know then that this early life mild disruption by this low dose of antibiotics depletes lactobacillus and SFB from the small intestine microbiome. So we wanted to see whether this impacted the small intestinal epithelium of these mice. So to do that, we first, so just to think about the intestinal epithelium, we know that you have a small intestinal stem cell pool, which is highly proliferative cells that would differentiate either in absorptive cells, such as enterocytes, or secretory progenitors, or secretory cells, such as goblet cells, Punnett cells, and Tuff cells. So we first wanted to see if we're altering intestinal epithelial proliferation. And to do that, we use a Ki-67 staining, and we also did EDU staining, and we saw the same thing, that animals receiving low dose of penicillin have a significant increase in intestinal epithelial proliferation, particularly in the stem cell compartment. We went on to do additional stainings, and this is immunofluorescence, in which green are secretory cells and red is enterocytes or absorptive cells. What we saw is that while in the mock-treated animals, they have significant numbers of secretory cells, these numbers are down in animals receiving antibiotics, and the opposite is true for the enterocyte population, suggesting there is a shift between the number of secretory and enterocytes in animals receiving antibiotics. We wanted to confirm this by doing single cell analysis in crypts and in epithelial cells from mock and low dose treated animals at 3 weeks. And this was the first time my lab did a single cell, so I was very excited that it worked. And what we saw with this is that, confirmed what we had seen by staining, is that animals receiving antibiotics, they have an increase in the proliferative compartment of the intestinal epithelial cells. and they have an increase in stem cells, transient amplifying cells, and a significant decrease in goblet cells, which is a secretory cell marker. And it's interesting, they also seem to have an increase in tough cells, but we don't know exactly how this is working so far. So since my lab is interested in nutrient uptake, we decided to look at whether there's a change in the capacity of these epithelial cells to absorb lipids. And the single cell data indicated to us that both immature and mature enterocytes in the animals receiving antibiotics here in dark blue, they have an increased lipid absorption and lipid metabolism capacity. And we've done additional analysis in the RNA-seq to confirm this. And we have some interesting genes that are upregulated in the enterocytes of the antibiotic-treated mice that we are following up on. But we really wanted to see whether these epithelial cells are really uptaking more fat. So to do that, we went back into our animal model, and we actually challenged these mice with BDP and in the presence of olive oil to actually be able to stain the chylomicrons and the epithelial cells taking up the fat. And so what we saw is that in this 21 days old or 3-week-old mice, it was actually very striking. So if you look in the mock animals here in green, you see the uptake of fat really in the tip of the villi. But if you look at the low dose treated animals, this uptake is significantly higher and it goes down more towards the crypts. And you can quantify this here. And you can see that results in an increase circulating triglycerides in the animals receiving antibiotics, really suggesting to us that this early life disruption of the microbiome is changing the gut microbiota, is changing the type of epithelial cells, so the differentiation of the epithelial cells, and increasing the lipid absorption. But we wanted to understand whether this has any impact in the host metabolism. So what we did, we went back to our animals, and now we looked at 3 weeks of age at the weight and adiposity. So what's important here is that we did not see any differences in weight and adiposity at 2 weeks of age when those animals have almost like no microbiome. But at 3 weeks of age, we did see that both male and female mice that received antibiotics weigh significantly more, and they have significant more abdominal fat, really showing that what we had seen in changes in the intestinal epithelium are mirroring and what we're seeing in overall growth and adiposity in the mice. So we wanted to see if this was really due to the microbiome. So what we did, we repeated these experiments now using germ-free breeders and exposing them to these antibiotics. And what we see is that in the absence of the microbiome, whether you expose pups to antibiotics or not, there's no difference. However, if you do a microbiota transplant in the moms, so if you restore the microbiome in the moms and then you expose them and the pups to the antibiotics, then we see what we had seen before, that really animals that receive antibiotics have increased abdominal fat and increased weight, so really implicating the microbiome in the phenotype. So we wanted to understand which microbes may be playing a role in this. So what we did, we went back to our germ-free mice, and this is all preliminary data. We're repeating these experiments. And what we did, we colonized the breeders with defined microbiota communities. So one of them was a community that had microbes plus SFB, which is one of the microbes of interest. The other one was a defined microbiome that had also lactobacillus. And what we saw is that actually SFB increased abdominal fat when compared to animals not having lactobacillus, not having SFB in the microbiome, but having lactobacillus. But when we also thought, since both of these microbes were depleted, we wanted to see what would happen if the mice had both microbes in the microbiome. And what we saw was very interesting is that if you have both microbes in your microbiome, you have decreased abdominal fat during early life, suggesting to us that these microbes may be interacting with each other, and both of them seem to be necessary to prevent this early life of increased adiposity that we're seeing when there's disruption of the microbiome. And we're performing some experiments now to try to figure out the mechanism behind it, how those two microbes are interacting with the host to prevent increased adiposity. So stay tuned. But lastly, what we wanted to see if this was long lasting, if these animals that had increased adiposity during early life, if they would continue to do that later in life. So what we did, we removed the antibiotics at 3 weeks of age, and we exposed the animals to either a high fat, we exposed either the animals that had never seen antibiotics and the animals that had received antibiotics to a high fat diet until the 8 weeks of age. And what we saw in the background of a high fat diet is that only male mice continued to gain more weight and have increased adiposity, and the females were protected. And we don't know the mechanisms yet of this sex-dependent phenotype. But what is interesting, is different from my first story, is that I don't have the data here, but we also see that this happens even if the animals are exposed to a low-fat diet. So it seems like this early life exposure to antibiotics have more striking long-term effects than when the animals are exposed later in life. And one thing that was very interesting to us is that some of those changes in intestinal epithelium development were also long-lasting. So you can actually see here that these are 8-week-old mice, is that even 5 weeks after removal of the antibiotics, the animals that had received antibiotics continue to have a decrease in the secretory cell lineage, and an increase in enterocytes. So we're trying to understand why this effect is long-lasting. So what we think is happening here is that in a healthy gut during very early life, prior to weaning, you have a strong colonization right before weaning of specific microbes such as SFB and lactobacillus that are interacting with the intestinal epithelium to promote proper intestinal epithelial development and differentiation, and to also control nutrient absorption, decreasing lipid, decreasing adiposity. However, if there's a microbiota disruption, and in this case it's through antibiotics, the beneficial microbes are no longer present, and those signals that are required for proper intestinal epithelial development are also not there, leading to increased nutrient uptake by the intestinal epithelial cells, leading to nutrient absorption and long-lasting adiposity. So with that, I wanted to thank the people that have done the work, especially my extremely talented grad student, Katie Shelton, that led both of these stories, helped by others in the lab, and my funding sources. So thank you.